The concept of an epigenetic clock is a groundbreaking biological test that determines age by examining DNA methylation levels. Essentially, the degree to which methyl groups have built up in a subject's DNA molecule. Having known since the 1960s about DNA methylation level susceptibility to age, vast research has been conducted on the CpGs whose DNA methylation levels fluctuate with age. A notable breakthrough was accomplished in 2011 by a UCLA team comprising Sven Bocklandt, Steve Horvath, and Eric Villen, showcasing that DNA methylation levels could predict one's age within an accuracy span of 5.2 years. Another team at the University of California in San Diego, run by Trey Edeker and Kang Zhang, created the Hannum Epigenetic Clock in 2013, leveraging 71 markers to accurately estimate age based on blood methylation levels. The next significant innovation came in the form of Horvath's Epigenetic Clock, developed by Steve Horvath. This multi-tissue clock, derived from roughly 4 years' worth of public Illumina DNA methylation data and the right statistical methods, was a game changer, offering the same prediction algorithm for any DNA source within the organism, without requiring any adjustments. This consistent measurement method allowed for the first time a comparison of ages of different body parts using the same aging clock. The key innovation in Horvath's clock was later built upon, resulting in the development of the intrinsic epigenetic age acceleration, an estimator based on blood cellular composition. Further advancements were made with second-generation epigenetic clocks that incorporated not only genetic variables like DNA methylation, but also environmental factors like smoking habits and chronological age. Among these, phenolage and Grimmage clocks were developed, both improving age estimation while phenolage factors in chronological age. Grimmage integrates the risks of age and smoking, allowing it to outperform previous clocks in predicting mortality. The third generation of epigenetic clocks is currently being developed, focusing on cross-species applicability, particularly in mammals. They determine the age of tissues from all mammalian species by analyzing DNA cytosine methylation in highly conserved regions. These advancements contribute to the continuous development of age determination tools which could remarkably assist in forecasting certain diseases. In 2010, a comprehensive age model was conceptualized, merging conventional age theories and epigenetics. This was expanded upon by Horvath and Raja, who proposed an epigenetic clock theory. They proposed biological aging as an unintentional result of developmental and maintenance programs, leaving molecular traces that allow estimation of an organism's age based on DNA methylation. They believed the connection between these molecular processes and the deteriorating tissue function likely involves intracellular changes, leading to the loss of cellular identity, as well as changes in cell composition, like the decrease in functional somatic stem cell. On a molecular plane, DNA methylation age is an indicator of an ensemble of inherent aging processes which collaborate with other separate aging elements to detrimentally affect tissue function. Biological aging clocks and aging biomarkers are anticipated as valuable tools in biological research, considering age is a key characteristic in most organisms. Precise evaluations of biological age could be pivotal in recording the effects of therapeutic interventions, including rejuvenation methods and forensic applications. Despite their potential, these clocks can only assess the impact of interventions on future aging rates and not interventions affecting all ages at a singular time. The said clock is an age estimation method using 353 epigenetic markers on DNA. These measure the methylation of CpG dinucleotides in DNA. These predicted ages have unique characteristics, being nearly zero for embryonic and induced pluripotent stem cells, showing a strong correlation with cell passage number, presenting a highly inheritable measure of age acceleration, and being applicable also to chimpanzee tissue, a human analogy. In biological testing, the rapid ticking of the epigenetic clock during organism growth and subsequent cell division slows down to a steady tick after adulthood. Age 20. The phenomenon wherein DNA methylation age of blood predicts mortality later in life supports the possibility of shared roots for both phenomena. It is also noted that physical and mental capacity markers are connected to the epigenetic clock, with lower capabilities linking to accelerated age. The clock tends to underestimate the age of elderly individuals. Noteworthy is Horvath's epigenetic clock's wide range applicability to various tissues and cell types, making it a tool for identifying evidence of disease. Accelerated aging in tissues. The Horvath clock, specifically the IA variant, shows links to several aging-related genes. The story explores the science behind epigenetic clock prediction, a process that revolves around the assessment of DNA methylation patterns. The investigation focuses on numerous gene markers, including UPNA4 associated with nuclear transport, C46 controlling innate immune response, and APADE4 related to lipid transport and Alzheimer's disease. The procedure comprises of creating a weighted mean of 353 clock CPGs, which is later transformed into denom age through calibration. The epigenetic ticking rate is noticeably high until reaching maturity, eventually settling into a steady rhythm. To estimate denom age, Horvath utilized training data sets and a penalized regression model examining over 21,000 PG probes across 2 platforms. The resulting denom age is treated as an estimation of chronological age, with 193 out of 353 CPGs directly correlating with age, and the remaining inversely relating to it. Tools for this process are accessible online. The median margin of error for age estimation is 36 years. Variations, however, do occur according to DNA source and complexity of tissue samples. The efficacy of the epigenetic clock is notable, showcasing a high correlation coefficient of 0.96 with chronological age. Comparison with other biological clocks, such as telomere length and 16ing for a gene expression, emphasizes its superior accuracy. Discrepancies between DNA methylation age and chronological age point to the interesting concept of age acceleration. This biological aging metric suggests that tissue can age faster or slower than expected. The story primarily focuses on the weak yet substantial relationship between lifestyle choices and epigenetic age acceleration in blood. Factors such as a well-educated background, plant-rich diet, moderate alcohol intake, and regular exercise suggest a slower rate of epigenetic aging, despite the potential risks aligned with metabolic syndrome. Interestingly, excessive intake of alcohol appears to quicken the aging of some. Epigenetic clocks. High body mass index, BMI, was analyzed in relation to the DNA methylation ages of various human tissues. There was a notable connection between BMI and accelerated epigenetic age in the liver. However, a minor but statistically significant connection was seen between BMI and inherent age acceleration in blood when a large blood sample size was studied. Moreover, metabolic syndrome biomarkers like glucose, insulin, and triglyceride levels, LHC reactive protein, and waist-to-hip ratio were identified as contributing factors to epigenetic age acceleration in blood. On the other hand, high amounts of high cholesterol showed links with a slower epigenetic aging in blood. Further research also supported the strong connection between high BMI, larger waist-to-hip ratio, and waist circumference and rapid epigenetic clocks. However, evidence also indicates that regular physical activity can minimize these effects. In cancer studies, denom age in women's breast tissues adjacent to cancerous tissue showed a higher than usual rate of acceleration. This suggests these tissues age faster than other parts of the body. Investigations also pointed out that epigenetic aging in cancer-free women's blood samples showed a faster rate way before diagnosis, indicating it as an early sign of cancer risk. Regarding cancer tissues, mixed effects were observed. Most types showed no significant link between age acceleration and tumor characteristics. However, cancers with T3 mutation had less acceleration than those without it. Divertingly, cancers with higher age acceleration had fewer genetic mutations in cases of breast and colorectal cancer. Certain genetic mutations were linked to amplified denom age. Epigenetic age of blood tissue could also potentially predict lung cancer occurrence. Findings have shown a link between accelerated epigenetic age in the human prefrontal cortex and various indicators of Alzheimer's disease, including a decline in overall cognitive and memory function. In older adults, the epigenetic age of blood also appears linked to cognitive function. These results underscore the potential of the epigenetic clock as a tool for measuring the biological age of the brain. Due to the lack of age-related biomarkers, it has been challenging to identify tissues that show signs of aging. However, by utilizing the epigenetic clock on 30 anatomical sites in both centenarians and younger individuals, it was revealed that the cerebellum ages more slowly and is approximately 15 years younger in a centenarian, which may explain its fewer signs of age-related dementia compared to other brain regions. In subjects younger than 70, the ages across brain regions and cells tend to be fairly consistent. Certain SNPs and genes relating to the epigenetic age of the cerebellum have also been identified. Huntington's disease is known to hasten the epigenetic aging in various human brain regions. Interestingly, semi-supercentenarians, individuals aged 105, 109, and their descendants exhibit a lower epigenetic age compared to their actual age. HIV-1 infection, while often linked to premature aging symptoms, has been challenging to detect at a molecular level. However, an epigenetic clock analysis has uncovered significant age acceleration effects in the brain and blood tissue of HIV-1 patients, echoing previous studies. Blood samples from Parkinson's disease patients, particularly granulocyte ratios, exhibit minor signs of premature aging. Children with the incredibly rare syndrome X who display toddler-like features throughout their life don't present a younger than expected blood tissue age when analyzed using an epigenetic clock. One of the perplexing aspects of biological aging revolves around the role and nuances of senescent cells. Of particular focus are the three primary categories of cellular senescence: replicative senescence, oncogene-induced senescence, and DNA damage-induced senescence. The ambiguity lies in whether they are all variants of a shared phenomenon triggered by diverse sources or whether they are distinct entities and how they relate to epigenetic aging. Interestingly, replicative and oncogene-induced senescence have been found to accompany epigenetic aging in primary cells, while DNA damage-induced senescence isn't the case. This separation of cellular senescence and epigenetic aging is reaffirmed by the observation of telomerase, immortalized cells still aging in line with the epigenetic clock, even without exposure to senescence triggers or DNA damage. In light of this, cellular senescence seems to be a state that cells are compelled into as a result of external pressures, including DNA damage, ectopic oncogene expression, and exhaustive cell proliferation. These senescent cells could ultimately cause tissue deterioration, leading to the physical manifestation of organism aging. However, at the cellular level, aging according to the epigenetic clock stands distinct from senescence. This intrinsic cellular aging process exists from the birth of cells and continues irrespective of the external pressures mentioned earlier. Additionally, the rate of aging varies between sexes and races. Male aging happens faster than female aging based on epigenetic age acceleration in various body substances. However, these rates are dependent on the specific body structure being studied and individual lifestyle. Similarly, ethnicity is linked with epigenetic age acceleration. The blood of Hispanics and Semonics in Semonage is slower, potentially explaining the Hispanic mortality paradox. Lastly, rejuvenating effects can be witnessed in blood due to stem cell transplantation. Hematopoietic stem cell transplantation, for instance, rejuvenates the epigenetic age of blood to that of the donor. However, this also comes with increased DNA methylation age linked to graft, versus host disease. Adult progeria, or Werner syndrome, has also been found to be associated with accelerated epigenetic aging in blood. The age-related changes in our body, though accurately estimated through biomarkers in our DNA, remain mystery. Why exactly we age is yet unanswered, and we still don't comprehend the real workings of the soul. Called epigenetic clock. A probable way to understand this is by comparing the readings of the epigenetic clock with the aging clock of our transcriptome. Various theories have been proposed, with Horvath suggesting that the clock is formed by a methylation footprint, a result of an epigenomic maintenance system. Cells regularly sustain DNA damage with about 50 double-strand DNA breaks per cell cycle and 10,000 oxidative damages every day. This damage repair can cause epigenetic alterations, and some of these changes, particularly increased methylation of CpG island promoters, persist even after the repair is done. Interestingly, similar temporary changes were noticed during the repair of damages caused by H22. Sometimes, these too might stick around post-repair. This buildup of epigenetic alterations can possibly contribute to the epigenetic clock. Moreover, it might simultaneously correspond to the buildup of unrepaired DNA damages, an element proposed to cause aging. Weidner et al. 2014 created a DNA age estimator focusing on 3 CpG sites of genes least affected by aging. However, this estimator is only applicable to blood and is less precise than Horvath's clock, yet its usage is cost-effective, making it a good alternative for some. On the other hand, Hannum et al. 2013, introduced several age estimators, each for a specific tissue type. These estimators also require extra data like gender, body mass index, or BAT. However, when these estimators are applied on different tissues, they lead to large errors due to poor calibration, calling for further refining in their model. The research underscores a key element of cancer, notably uterine corpus endometrial and triple-negative breast cancer. The age of cancer tissue is often younger than anticipated. In assessing age, Hannum's method of estimating uses information including body mass index, gender, diabetes status, ethnicity, and batch. While this method holds value, it falls short when applied to new data due to differing batches and potential for large errors. In a bid to corroborate this perspective, Giuliani et al. Explored the significance of DNA methylation levels in determining age through the genes in human teeth. One area of interest to the researchers is in historical and ancient teeth. Meanwhile, Galkin and his team have harnessed the power of deep neural networks to create an age predictor tool of unmatched precision, using in excess of 6,000 blood samples. The team plans to make this tool, which can offer age predictions for those with conditions like obesity, ovarian cancer, frontotemporal dementia, and EBD, widely accessible in 2021. Extensive research conducted by 18 different groups across 3 continents indicates that DNA methylation-based epigenetic tests are ready for comprehensive clinical use. Further research by McCartney et al. Proposes that plasminogen activator inhibitor 1 or PAON is another potential tool for age prediction, particularly when considering its strong correlations with cardiometabolic diseases. Furthermore, the robust pan-tissue epigenetic clock, Altimage, created by De Lima Camilo et al., using an optimized deep neural network, has shown exquisite precision in age prediction. It outpaces Horvath's linear method due to its ability to identify CpG interactions. Interestingly, unlike Horvath's clock, Altimage calculates the age of a cancer to be older than normal tissue. Lastly, studies in mice by Wang et al. And Petkovich et al. Have found that mice undergoing lifespan extending protocols such as calorie restrictions showcase a younger epigenetic age as compared to their untreated counterpart. Age prediction in mice based on DNA methylation has been refined to reach an error of less than 4 weeks.